biotin scavenging protein. But in this case, they're just using the strep avidin to bind to the biotin. And attached to the strep avidin is an actin filament. Right? And this actin filament is long enough that you can see it under an electron microscope. Okay? So they hooked this on to a reconstituted ATP synthase system. Put in hydrogen, put in an acidic solution so that hydrogen ions would try to flow through. Gave it ADP so that it could act, ADP and phosphate so that it could act. And this is what they found, right? This is that actin filament. Look what it's doing. What's it doing? It's rotating. Yeah. If you follow it, that filament is whipping around like a propeller. Right? Here, 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 there, 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 there. Right? It goes up, now it's this way, this way, this way, this way. Right? It's rotating. So as hydrogen ions are flowing in, this thing is rotating. Right? And so as this is twisting, these subunits, as they change their relationship to this A and B, they are changing their conformation. And their conformation is changing such that when they reach a particular conformation, they catalyze the formation of ATP from ADP. Now, I can't tell you exactly what the chemistry is of that, but this creates enough of an energetic potential within these subunits to create a microenvironment where the formation of ATP becomes favorable within that enzyme. Yeah?
So the subunits have a conformational change?
Mm-hmm. Yeah. Right? This alpha-beta dimer has a conformational change that within that microenvironment created, the formation of ATP becomes favorable. Now, this is kind of cool. I mean, if you think about it. So they looked for things that were similar to this, right? At first they thought, it's something unique, something related to ATP synthesis. But they started looking around after they noticed this movement, or actually they had found this similarity, which is what made them look for movement. What kinds of proteins are most similar to these? The proteins of flagella in bacteria. Right? This is essentially a molecular motor that our mitochondria have learned to run backwards. And we can show that. If we give it ATP and don't create a hydrogen ion gradient, then ATP causes this to rotate in the other direction, and hydrogen ions flow from the matrix side to the intermembrane space side. So what we do is we just push everything backwards. We use up our ATP, so we have lots of ADP and phosphate here, but not a lot of ATP. And we actively pump hydrogen ions out in another way, so that hydrogen ions come back in backwards, push the motor to run backwards, which makes it synthesize ATP instead of breaking up ATP to ATP. That's kind of cool. it's, these proteins are most closely associated with the anchor proteins, what's it called? There's a word for it, but I'm forgetting in biology. Probably they haven't talked about it since general biology, or if you took microbiology. There's a word for the proteins that are at the base of a flagellum, that are the integral proteins in a mitochondria. Those are the proteins that these are most closely related to. Nobody's frowning.
No, I'm trying to remember.
You're trying to remember the word? There's a word.
Aren't they the groups of 9 cilia things?
Mm-hmm. There's a word for that one. Yeah. But anyway, right? So essentially, our mitochondria are taking proteins that evolved. I usually avoid these kinds of conversations, but we'll go with it today. It evolved as motor proteins to drive flagella. And they've learned to run them backwards. Just like an electrical motor, right? If you hook up a battery to an electrical motor, it will turn in one direction. If you grab the leads of that same motor and turn it rapidly in the opposite direction, what happens?
It gets mad.
It'll shock you, right? A molecular motor running backwards becomes a generator. Right? So that's what we do. We run our molecular motor backwards. We use hydrogen ions flowing in to run it in the opposite direction. And that then we can use to generate ATP energy. Sarah looks impressed by that.
I was just thinking about how we're all this backwards bacteria now.
That's what the bacteria think, but that's okay. Okay? That make sense? Any questions about that? Okay, some other stuff, interesting things related to studying this. And one of the points they made is that if you isolate just some of the mitochondrial inner membrane, right? If you just isolate a piece of the mitochondrial inner membrane with the complexes present, right? The complexes will oxidize in ADH. They will pump hydrogen ions as long as Q is present. They will reduce oxygen, which takes more hydrogen ions out of solution. But they won't make ATP. Why? That's okay.
I mean, do we give it ATP?
Sure. If you have ADP and phosphate, right? If you isolate that membrane and have it there, here's the key. Have it there as membrane fragments, not as intact membrane. If you give it in ADH, it will oxidize that in ADH, but it won't make ATP. Why?
Can't create the gradient.
Why can't you create the gradient?
Because it'll just like diffuse out and kind of just flow back.
Right.
To wherever.
We can pump hydrogen ions, but if the membrane isn't intact, those will take another easier path to diffuse back into the other space. So you need the intact membrane to drive those hydrogen ions through that ATP synthase. Right? So if your membrane isn't intact, you get no ATP synthesis. That's good when you can get a Rolling Stones callback into the biochemistry class. Okay? Now, let's see. Let's go to this figure. This is a compound called 2,4-dinitrophenol. Right? 2,4-dinitrophenol. 2,4-dinitrophenol, something that's easy to make. We could make it in organic chemistry lab easily with a nitration reaction. Really easy to make. 2,4-dinitrophenol. One, it's got this organic aromatic ring, so it tends to be soluble in membrane bilayer systems and diffuse across them rather easily. And it is a phenol, so it has, right, this hydroxyl on the benzene ring can be fairly easily deprotonated. When you add the nitro groups, the pKa comes down to, right, you add electron withdrawing groups to the ring. Normally a phenol has a pKa around 10. So this one probably has a pKa around 8, I would guess. I'd have to look it up. But it's more acidic than phenol normally is. And so it can be deprotonated. Well, what's the approximate pH of the cytoplasm?
7.4.
7.4, right? So at if this has a pKa somewhere between 7 and 8, a substantial part of this is going to be deprotonated, right? But any that is close to the mitochondrion, because hydrogen ions are being actively pumped out of the mitochondrial matrix, you get a microenvironment near the mitochondrion of lower pH, higher hydrogen ion concentration. So any of this that's in there can be protonated, right? But once it's protonated, it's likely to diffuse across that mitochondrial inner membrane. Actually, my key is this way, so it should be this way, right? So you've got deprotonated in the cytoplasm. We pump hydrogen ions out of the matrix, just like we were talking about, create a microenvironment of lower pH, higher hydrogen ion concentration, which tends to protonate the 2,4-dinitrophenol. That 2,4-dinitrophenol can then diffuse into the matrix, taking the hydrogen ions with it. But since we pumped hydrogen ions out of the matrix is more basic than this space was. And so that tends to deprotonate when it gets into the matrix, which tends to then